We've already gone over photosynthesis, and I just wanted to do a little recap before we head into cellular respiration in this lecture. But in photosynthesis, it was everything happening inside of plants, inside chloroplasts, where we have water going in, we have carbon dioxide going in, and through light reactions in the Krebs cycle, we're able to get out oxygen and carbohydrates. And if you recall, carbohydrates always have a ratio of 1 carbon to 2 hydrogen to 1 oxygen. In cellular respiration, we're going to take some of that carbohydrate, and our example is going to be glucose, which is C6H12O6. It still has that 1 to 2 to 1 ratio, and we're going to take that oxygen that we got out in photosynthesis, and we're going to head inside of an organelle called the mitochondria and do cellular respiration. And the outcome is going to be carbon dioxide and water, those two things that need to go right back into photosynthesis. So we're going to take that equation and almost reverse it. So cellular respiration is going to happen in animal cells, any cells that have a mitochondria. And I know there's a lot of wording on this slide, but I just wanted to go over some kind of key topics, and then we're going to break this down. So cellular respiration is a cellular process. It breaks down nutrient molecules such as carbohydrates that were produced by photosynthesizers. These were carbohydrates that were made from those plants through photosynthesis with the production of ATP. So we're going to generate ATP at the end. It consumes oxygen and it produces carbon dioxide. So it uses oxygen that plants gave us and it produces carbon dioxide that plants need. It is an aerobic process because some of these reactions that are happening do require oxygen. And it usually involves a complete breakdown of glucose, that's C6H12O6, all the way down to carbon dioxide and water, things that can then go into photosynthesis. Energy, that ATP, is extracted from the glucose. We know that sugar has energy in it, but we're going to break it down to really try to get out all the energy that is in there. So it's released stepwise, and it allows ATP to be made as efficiently as possible. Now we are going to have some oxidation reduction reactions and enzymes, and some that are going to be predominantly found in cellular respiration are NAD plus and FAD. These, as well as some coenzymes, are going to be key players as electron carriers. So a little recall on what are reduction oxidation reactions, also known as redox reactions. So electrons are removed from substrates, and again, we can have, anytime we are gaining electrons, it becomes reduced. If you are losing electrons, it becomes oxidized. If that oil rig acronym of oxidation involves loss and reduction involves gain, and it's because you're gaining something that's negative. But as you are gaining electrons, you are gaining some hydrogen as well, because those electrons are usually tied to an element. And so our oxygen is going to become reduced and make water as an outcome. And it's becoming reduced, it's gaining electrons, but it's also gaining some of that hydrogen. Oxidation is losing electrons, so it's actually, if you notice, you're going to lose the hydrogen here, and so it's losing electrons. So glucose becomes oxidized, it's losing the electrons, and your oxygen is becoming reduced, it's gaining some of those electrons, and again, with it, those electrons are tied to water. So we care quite a bit today in this lecture about those electron carriers. What is able to easily pick up electrons, and with it some hydrogens, but it's also carrying energy. Those electrons do have energy. And a couple key players that are really able to pick up those electrons and pick up those extra hydrogens with it are NAD plus and FAD. So NAD, and I am not going to require that you know what it stands for, but I do have it written there in case you were curious. These two molecules easily pick up and drop off electrons and then the hydrogens with it. NAD plus easily picks up 1 electron and 1 hydrogen, and FAD can easily pick up 2 electrons with 2 hydrogens. Because it can pick up 2 electrons, it is a more efficient electron carrier. It can just pick up more electrons, carrying more energy with it. The whole cellular respiration has 4 phases to it, and we're going to go through and discuss what's happening in each of the phases. And as I said before, the breakdown of glucose to harvest all that energy out is a stepwise process. And if we can get through all 4 of these steps, we're going to generate the most ATP in the most efficient manner. So the first step is the glycolysis step, which happens out in the cytoplasm. So it's not happening in that mitochondria yet. And it's breaking down glucose, and it doesn't need any oxygen yet. Then we have what's known as the preparatory or prep reaction, which is now happening inside of the mitochondria. And what we get out of glycolysis gets further broken down. We have the citric acid cycle that's happening in the mitochondrial matrix, which is really just the fluid inside of the mitochondria itself. And it's where we're going to get a release of a lot of carbons from the breakdown of our carbon glucose molecule. And we're also going to start to get a couple ATP. And then we have our final electron transport chain. Again, it's why we care about those NADs and FADs, because they're picking up all those electrons and hydrogens with it. And this is happening on the mitochondrial membrane, on some of these inner folds in the mitochondria, and it's really where we generate the maximum, the highest number of ATP. So we do get a little ATP out of glycolysis, we get a little of ATP out of the citric acid cycle, and we get a lot of ATP out of the electron transport chain. Here's a bigger picture of it, just showing those breakdowns, your 4 steps that are going to be able to take glucose and ultimately make lots of ATP, lots of energy for our cells. Another picture showing it, just depends on different books. Some people like different things. I like to present things in a lot of different ways. I also love pictures on PowerPoints because I truly do think that pictures speak 1,000 words. So just showing what's happening in glycolysis and where, our prep reaction, what's happening, our citric acid cycle, and where it's happening in the electron transport chain. And then again, where we're getting our ATP out. So we're first again going to start with glycolysis, which is happening in the cytoplasm or cytosol of the cell. So it's happening just in our cells. It's not happening in the mitochondria yet. And the word glycolysis means, lysis means to break apart, glyco means sugar. So it's in this step we're going to take that carbohydrate, that glucose, and we're going to break it down. And we separate glycolysis, although it is our first step in cellular respiration, there's really kind of 2 parts that are happening in glycolysis. We have an energy investment step and we have an energy harvesting step. If you recall, in chemical reactions, we need some activation energy to start reactions off. So before we can start cellular respiration, we need to put a little bit of energy in just to get this thing going, and that's where we have 2 ATP. So before we make any ATP, we actually have to put a couple ATP in just to get this reaction going. And what that little bit of ATP is going to do, it's going to split glucose into 2. So it's going to evenly divide it into 2. And it's going to make 2 molecules called G3P. In the energy harvesting step, that's where we're going to take that G3P that we just made and really break it down. And it's anytime we break this down, we're releasing a little bit of energy and we're getting something out of it. So we're going to remove electrons, and with it the hydrogens that go with it. We're going to find an electron acceptor that's going to pick those up. So we're going to get some NADHs, got now full electron carriers. We're going to get 4 ATP by substrate level phosphorylation. It just means we're going to add some phosphate onto ADP. And so by the end of glycolysis, we've made a net of 2 ATP. So we made 4 ATP, but it took us 2 ATP at the beginning. So we made 4, but we used 2. So we've really only made or generated a total of 2 usable ATP. And those G3Ps that we made at the end of that energy investment step turn into a molecule called pyruvates. Now again, I like pictures, so kind of showing what's happening. And I'm not going to require that you know every single intermediate step. Just kind of know, what do we get out of glycolysis? What goes in, what do we get out? So what we what goes in is we have our glucose is going to go in. And again, right away, we've got some ATP that's going to go in. That's your activation energy just to get this reaction going. And that ATP is going to split the 6-carbon molecule, the 6-carbon glucose, into 2 3-carbon molecules. Again, we haven't lost any carbon. All we've done is split this 6-carbon molecule into 2 3-carbon molecules. We've at this point used 2 ATP, but we're going to further break that G3P down. And we're going to look to see what are some things we get out. And so we get out some NADH, one on each side. We get out some ATP here. We get out some more ATP here. And ultimately, we make a final product at the end of glycolysis of pyruvate. So we put in glucose. We get out 2 pyruvates. We still have 6 carbons. We haven't lost any carbons yet. We've made 4 ATP, but it costs us 2 ATP to do that. So we've only gained 2 ATP. But we did get a couple NADHs. We've got 2 NADHs here. Just showing in glycolysis, we have our glucose molecule that through the glycolysis, through all those little tiny steps, we are going to gain 2 NADHs, we're going to gain 4 ATP, but it cost us 2 to do that. So we really only gained a net of 2, and ultimately we get 2 pyruvate molecules. Another picture or review of just what goes in, what comes out of glycolysis. So what went in was a 6-carbon glucose. What came out was 2 3-carbon pyruvates. Again, still a total of 6 carbon. We have not lost any carbon yet. 2 NAD pluses went in. We get a couple NADHs out. They have now picked up electrons and picked up hydrogens. And they're going to head off to the electron transport chain, which is our last step of cellular respiration. We did put in 2 ATP, which turned into 2 ADP, and then we put 4 ADP in, plus some phosphorus, and with the energy we got out of breaking down glucose, we were able to take those ADPs and phosphates and turn it into 4 ATP. So we've netted 2 ATP. Not a lot. So then the question then comes is, well, what do we do next? As I said before, cellular respiration is an aerobic, oxygen requiring chemical process, but we can do glycolysis. Oxygen wasn't involved in any of glycolysis up until this point. So the question becomes is, well, what if we don't have any oxygen? Does it stop? Like, do we just take these pyruvates and we're done? It doesn't. There are things where this pyruvate can go depending on whether there's oxygen or not. So we're first going to talk about, well, what happens if there isn't oxygen? What can this pyruvate do if there is no oxygen? So no oxygen, no problem. Pyruvate is still a metabolite. It's still a chemical that can go and do cellular respiration, but it can also get broken down as well and provide other products. So if oxygen's available to the cell, it's going to head to the mitochondria and continue on with aerobic respiration, which we will continue on as well and follow what happens. But if oxygen's not available to the cell for whatever reason, it's just not getting enough oxygen that it requires, that pyruvate can go through what's known as fermentation. It's an anaerobic process, so it doesn't require any oxygen, and it happens in the cytoplasm. So it's not going into any particular organelle. It still all happens out in that cytosol or cytoplasm of the cell. So glucose can, without any oxygen present, turn into lactate, depending on what the organism or what cell it is that's breaking down glucose, or it can turn it into ethanol and carbon dioxide. My fun fact, ethanol and carbon dioxide is released when you are making bread. So yeast can take sugar and they can break that sugar down without any oxygen present through fermentation and make ethanol and carbon dioxide. And usually I have a student like, so there's alcohol in my bread? There is before you bake it. Once you bake it, you bake all the alcohol off. But it's that carbon dioxide that's getting released from those yeast that actually makes the bread rise. It's the air bubbles that it's producing that makes your bread rise. And then you bake off all the ethanol when you bake it. It kind of gives it that bready smell if you've ever made bread. But Some organisms can, whether it's yeast or some other type of cell, can also take that pyruvate and they can turn it into other products as well. It doesn't have to be just lactate or just ethanol. And it's those NADHs that can transfer all their electrons to the pyruvate. They can take that pyruvate, they can give it electrons and help turn it into a different type of product. This is just showing some examples of what can we get out of pyruvate in a fermentation reaction. So certain things like yeast can do an alcoholic fermentation. That's where we can get our ethyl alcohol and carbon dioxide as a byproduct. There are some things like some bacteria, some fungi that, including some of our own cells, that can do what's known as lactic acid fermentation, that can take sugar and they can make it into lactic acid. And then other organisms, a lot of different types of bacteria, can take that pyruvic acid and turn it into other things as well. And this is showing in examples. If you are Propionibacterium, you can take pyruvic acid and turn it into propionic acid and carbon dioxide, which is what gives Swiss cheese its kind of sharp flavor, but it's that carbon dioxide that it's producing that actually makes the holes in your Swiss cheese. Aspergillus, Lactobacillus, and Streptococcus, all bacteria, can take pyruvic acid and make it into lactic acid, which gives a sharp flavor to cheddar cheeses, to yogurt, and to soy sauce. Saccharomyces, that's your yeast, can turn pyruvic acid into ethanol and carbon dioxide. The bacteria called Clostridium can take pyruvic acid and can make your isopropyl alcohol and your acetone that's found in your nail polish remover. So different organisms can take pyruvic acid and still break it down, and this all is happening without any input of oxygen. Why fermentation is good, but why fermentation is also not so good. So fermentation is good because it does still provide a quick burst of ATP. So fermentation still requires the breakdown of that glucose. It still has to go through glycolysis to yield that pyruvic acid. And then just what do we get out of pyruvic acid? That's a little bit different. But through glycolysis, as we already went through, I'm going to go back a couple slides, we do get out a little bit of ATP out of glycolysis. So we do get out a little bit of ATP. We don't get any ATP out of breaking down pyruvic acid, but through the whole fermentation process, we do get a little ATP out of the glycolysis step. So there are times we just need some really quick burst of ATP. It's not a lot of ATP, but we need a quick burst of it. The not so good part of the fermentation, that lactate or lactic acid or an alcohol that could be made, is toxic to cells. It's why if you do certain types of exercises and you are sore the next day, is that your muscles were working because you told them to work, but they didn't have enough oxygen to break down glucose efficiently, so they had to go through fermentation. They made lactic acid, and that acid is toxic to your cells, and it causes your muscles to fatigue and feel sore the next day. Now, if you're yeast, they actually die from the alcohol they made. So the yeast will happily make some alcohol and break down all that glucose, but then they actually make enough alcohol that it kills them. So they make a product that ultimately kills them. So good, but there's side effects as well. Now, the efficiency of fermentation is not great. Again, it gives a quick burst of ATP, but it's not the most efficient way to break down glucose and get all the energy out of glucose that there is there. One molecule of glucose can yield 2 ATP through that glycolysis step, and it's equivalent to about 14.6 kilocalories. We just call those calories. If you could, with oxygen present, fully break down glucose, you could yield 686 kilocalories of energy. So we can get a little bit of energy out of the fermentation step, and that's really coming from the glycolysis step, but it's not that huge, large number of energy that we could get out if oxygen was present and we could go through that whole cellular respiration process. So we only get 2 molecules of ATP versus 36 to 38 if we finish the whole reaction. So it gives us our quick energy, it's just not a lot of energy. So we're going to continue it now if there was oxygen present. So at the end of glycolysis, we made pyruvate, also known as pyruvic acid. Different books call it different things. And that pyruvic acid goes into our next step, our second out of the 4 steps, called the prep reaction or preparatory reaction, which happens inside the mitochondria. It's just a reaction that connects glycolysis, our step 1, to the citric acid cycle, our step 3. So we take our end products of glycolysis, our pyruvate, it's going to go into the mitochondrial matrix. That's the fluid inside of the mitochondria, and that pyruvate is going to get converted to a 2-carbon acetyl group and attached to something known as coenzyme A. So what we're going to get put in and what we're going to get out. So we did get 2 molecules of pyruvate out of the end of glycolysis, but we're just following one of them through. So at the end of glycolysis, we have pyruvate. So we're going to take pyruvate, we're going to turn it into acetyl coenzyme A. What we're going to get out is we are going to get an electron carrier, the NADH, out of every pyruvate we have. We're also going to get some carbon dioxide out. It's where does that carbon come from? So pyruvate is a 3-carbon molecule, but acetyl-CoA is a 2-carbon molecule. So the carbon doesn't magically disappear. Instead, that carbon is lost as carbon dioxide. I'm going to take our final product here, our acetyl-CoA, and again, we had 2 pyruvates at the end of glycolysis. We're just going to follow one of them through. So just note there's this pyruvate, but there is another pyruvate as well that's going to do the exact same thing. So ultimately we would have gotten 2 carbon dioxide, 2 NADHs, and 2 acetyl coenzymes A out of our 1 molecule of glucose. But we're going to take our acetyl-CoA and it's going to go into something known as the citric acid cycle, which is still inside the mitochondrial matrix, that fluid, also called the Krebs cycle. And it begins with that acetyl-CoA going in, and it's going to combine with a 4-carbon molecule called oxaloacetate. You do not have to know how to spell that, maybe just kind of recognize it as that it's found in that citric acid cycle. And it's going to form a 6-carbon molecule called citric acid, where this whole cycle gets its name. So we have a 2-carbon molecule plus a 4-carbon molecule, yielding us our 6-carbon molecule. We're also going to have NADH and FADH2 that are going to be capturing lots of electrons and head off to our electron transport chain. We're also going to get a couple ATP out of here as well. And my reminder that the citric acid cycle, it's going to happen twice. They say it turns twice, because 1 molecule of glucose yielded us 2 pyruvate, which yielded us 2 acetyl-CoAs, and so each acetyl-CoA is going to go in to the citric acid cycle. So ultimately, at the end of the citric acid cycle, we're going to get 4 carbon dioxide, 2 ATP, 6 NADHs, and 2 FADH2s. We have a ton of electron carriers that are coming out of the citric acid cycle. We're also, at this point, we had a 6-carbon glucose molecule. It was C6H12O6. We lost 2 of them when the pyruvate turned into acetyl-CoA, and now we're losing the other 4 carbon. So at the end of the citric acid cycle, all 6 of the original carbon in glucose have now been lost as carbon dioxide. We don't get a lot of ATP out of this step, but the biggest things we really get out of here are these electron carriers. So what this looks like as a cycle, we have our 2-carbon acetyl-CoA that's going to combine with our 4-carbon oxaloacetate, and it's going to turn into that citric acid, which is a 6-carbon molecule. Now, as that citrate or citric acid starts to break down into different intermediate things, this is where we start losing carbon dioxide. We lose a carbon dioxide here, we lose a carbon dioxide here, so your 6-carbon goes back to a 4-carbon. Just as a reminder, this cycle happens twice. So ultimately we lose 1 carbon here, 1 carbon here, and then as it goes through a second time with our second acetyl-CoA, we lose 2 more carbons as carbon dioxide. We also get out, we have an NADH here, an NADH here, an NADH here, so 3 of them, but times 2 because we've got 2 turns of our citric acid cycle. And we have our FADH, which we again going to turn twice, so we're going to get 2 of them. So we get lots of electron carriers, and we get 1 ATP out of each cycle. Just another picture showing the Krebs cycle. We have our 2-carbon acetyl-CoA mixing with a 4-carbon oxaloacetate or oxaloacetic acid. Makes our citric acid, break it down, we lose carbon dioxide here, we get some NADHs at 3 different places, as well as the FADH or FADH2, and our ATP. And just to recall, one glucose molecule does turn this twice, so we kind of double everything that comes out. So just as a review of what's going in and what's coming out of the citric acid cycle. So we do have 2 acetyl-CoAs going in. We have some NADHs that will turn in, or NADs that will turn into NADHs, some FADs that will turn into FADH2s once they've picked up those electrons and the hydrogens that go with it. And we had a couple ADPs that turn into ATP. So we've got all of our carbon has now been lost as carbon dioxide. We've got lots of electron carriers, and we have ATP. So we head into our 4th and final step of cellular respiration, the electron transport chain. So all those electron carriers are going to finally head here and do something. So in a eukaryotic cell, a more advanced cell, this is all going to happen on the inner folds. So this inner membrane of the mitochondria. A prokaryotic cell, such as those that bacteria have, mitochondria are just way too advanced of an organelle. They don't have mitochondria. So this electron transport chain all happens on their outer plasma membrane. It is a series of carrier molecules that are moving things from one side of the membrane to the other side of the membrane. And again, pictures are going to come up and it'll help explain it a little bit more. But the electron transport chain is where all those electron carriers went. So any electron carriers that we got out of any of our steps along the way, whether it was NADH or FADH2, it's going to go towards the electron transport chain. And it's these electrons and the hydrogens that they have that are going to allow us to generate lots of ATP. And as we said before, this is an aerobic process. And so those hydrogens do have to eventually land somewhere. And they will find an oxygen as a final electron acceptor. And when you mix hydrogens and waters together, you form water, which is one of our outcomes of the cellular respiration process. So what happens to all that NADH? What happens to the hydrogens that are attached, and what happens to the FADH2s? So we do have, again, here's our membrane. This is that inner membrane on the mitochondria. And we have our NADHs or our FADH2s that are bringing their energy, they're bringing their electrons and their hydrogen with them, and ultimately they're going to make ATP. But I'm going to break down a little bit on how that happens on my next slide. NADH has enough energy in it to make 3 ATPs. So one NADH can make 3 ATPs. FADH2s only have enough energy to make 2 ATPs. But either way, we're making a lot of ATPs just based on one electron carrier. So same picture. So what are those electron carriers doing? So they have energy in them, and they have those hydrogens attached as well. They're going to use the energy from the electrons to pump the hydrogens across the membrane. So all of a sudden we have a whole bunch of hydrogens on one side of the membrane. They're pretty crowded. I know this picture only shows 3, but they are quite crowded. And they don't like to be crowded, so what they want to try to do is they want to sneak back to the other side where there's not as many hydrogens. So they will find an enzyme called ATP synthase. And as a hydrogen molecule sneaks through this particular enzyme, it turns the enzyme, and kind of like a turbine, it generates energy. So as this enzyme turns, it takes the energy that occurred from turning, and it can attach that phosphate onto ADP, and it makes ATP. So we've taken the energy from these electrons, pumped hydrogens across the membrane, made them so they were unhappy because they were so crowded, so that they want to sneak back through this particular enzyme, and as they do it, it spins the turbine and generates ATP. So kind of breaking down what I have on here, we have a bunch of hydrogen ions that are all pumped across the membrane, and they have a charge to it. They are positively charged, but they are very crowded and they don't like it. And so they want to go where there's not as many. And so that means they're going to flow down their gradient, meaning going from a high concentrated area to a low concentrated area. And when they do that, they go through that ATP synthase, and it's that enzyme tacks on that extra phosphate onto ADP to generate our ATP. So we're using all these energies, we're making hydrogens crowded, and then because they're crowded, they want to go somewhere where they're not so crowded, and they spin this enzyme and generate ATP. It's kind of our simplified version of it. But again, some of those hydrogens do have to end up somewhere. They can't float around just as hydrogen ions, and so they will eventually find some oxygen. And as they attach to oxygen, that's where we get water out. So this is another picture, different book showing that. I think it looks a little more confusing because I said, well, yes, there's lots of hydrogen ions. Simpler just to look at 3. Then, yes, there's lots of hydrogen ions. They're not happy. We pumped them all across here using the energy from those electrons, but they want to sneak back, so they can sneak back through this enzyme, ATP synthase, which turns ADP and phosphate into ATP. Again, some of those hydrogen ions at some point have to find some oxygen, and then they generate water. This, I personally just think it looks a little more confusing, but it's showing that same basic idea. So just accounting for all the energy that we've made from one glucose molecule. So out in the cytoplasm, one glucose molecule through glycolysis made us 2 ATP. Not a lot, but we got 2 ATP out of glycolysis. From pyruvate to acetyl-CoA, we didn't get any ATP, but we still needed to get that starting material of acetyl-CoA to go into the citric acid cycle. And from there, we got 2 ATP. Still not all that much. Between these 2 steps, we've generated a total of 4 ATP. But through these steps, we did get a lot of NADHs and FADH2s out. And from them, we generated a lot of ATP. Because if you recall, NADHs can usually yield 3 ATP per NADH, and FADH2s can generate 2 ATP from every FADH2. So ultimately, we get a total of between 32 and 34 ATP. Now, I usually have a question of why is there a difference? Well, different organisms and cells and mitochondria are sometimes a little more efficient at making to ATP or not, and so sometimes this can vary among the cell or among the organism itself. But ultimately, we make between 36 and 38 ATP. Most of it's in our electron transport chain, but we still have to add it to that original 4 ATP we get out of glycolysis and out of our Krebs cycle or citric acid cycle. So quite a bit of ATP. If we can go through the whole process of breaking down glucose in the presence of oxygen, so not fermentation, we can make 36 or 38 ATP. Fermentation alone only gets us 2 ATP. But where do we get all this glucose to even do all of this? Well, it's from food. So it's where we get all of our energy. It comes from carbohydrates. We have fats that can get broken down. We have proteins that can get broken down to generate these carbohydrates to eventually get fully broken down and generate all of this ATP. So anytime we are breaking down molecules, those are those catabolic reactions that are releasing energy. However, we can use the energy from the breakdown of products to also build molecules. So as we are breaking things down to release energy, we are also using that energy to build molecules that are required. So some of those intermediate things can actually be building blocks. Now there's a lot going on this particular picture that I have on here, and that's fine. I almost wanted it to look like there's a lot going on. All the purple arrows mean it's a breaking down pathway, a catabolic pathway. All the green arrows means we are a building molecule. So this is showing just an example in glycolysis. Although in glycolysis we take glucose, we make pyruvic acid, but there was all of these intermediate steps that occurred along the way. Well, those intermediate things that are made just to go from glucose to pyruvic acid, they can be used as building blocks. They can make lipids. They could make nucleotides or amino acids to eventually make proteins. So these different things can be a product, but they can also be a reactant, something that's going to go in and make something else. Just as an example, fats We can take G3P and make it into glycerol. We can take acetyl groups and make it into fatty acids. These products also become reactants. Now, my note about proteins. So a lot of these things can go in to make amino acids and ultimately make proteins that our cells need in various structures and enzymes, but not all amino acids can be made by our cells. There are 20 different amino acids out there, and our cells can only make 11 of them. So although, yes, it's showing we can make amino acids and we can make proteins, our cells can only make 11 of the 20 different amino acids, which means the other 9 different amino acids that are needed to make proteins, to make enzymes, they have to be part of our diet. We can't make them, our cells can't generate them, and so we have to get those amino acids from somewhere. And so we get them from eating different types of protein molecules that can get broken down to give us those amino acids as building blocks. So our cells can't make everything. We do have to get some things from our diet. As a nice summary, I started off with this slide as well. In photosynthesis, we took water, we took carbon dioxide, we're ultimately able to make oxygen and some type of sugar. In cellular respiration, we're going to take sugar and we're going to take the oxygen that we made and we're going to turn it into water and carbon dioxide. So the things that are going to go into photosynthesis are the things that come out of cellular respiration. And so it truly is this cycle that plants are generating the things that we need, and then we generate the things that plants need. Kind of everything all goes around, but they are opposite equations of each other.